The high-security kit is made of 3 parts. A high-security vitrification straw, transparent with a flared extremity and a pre-sealed extremity weighted with a stainless steel rod to prevent buoyancy when immersed in liquid nitrogen. A capillary tube with a gutter which is attached to a coloured handling rod. Colours include white, blue, red, yellow, purple or green and are weighted with a small metal tube in its central part. A blue plastic insertion device. Connect the blue insertion device to the coloured handling rod. Prepare the straw identification, making sure to use a cryogenic LN2 resistant label marked with permanent LN2 resistant ink. Apply the label at least 20 mm from the flared extremity of the straw. Protect the coloured rod and capillary connected to the insertion device by partially inserting the gutter inside the straw until the kit is actually going to be used. Prepare the biological sample for vitrification according to the instructions provided by the media manufacturer. Be sure to comply with recommended exposure times for the different solutions. When sample is ready to be vitrified, take the handling rod and connected capillary in one hand. Then bring the extremity of the gutter close to the drop of media containing the embryo or oocyte. Using a micropipette with the other hand, carefully take and deposit the microdrop at about 1 mm from the end of the capillary gutter. Insert the handling rod and capillary inside the HSV straw until the body of the blue insertion device comes in contact with the flared extremity of the straw. Slightly pinch the straw between your 2 fingers and remove the insertion device. Seal the open end of the HSV straw using a SIM sealer. Immediately immerse the HSV straw in a liquid nitrogen bath while making circular movements to prevent the formation of any insulating air layer. Transfer the HSV straw to its storage device. Identify the HSV kit to retrieve and transfer it into a separate dewar filled with liquid nitrogen near the proximity of a microscope. Prepare the thawing dilution solutions according to the instructions of the media manufacturer. Lift the straw enough to expose the coloured rod. Hold the coloured rod area of the straw with 2 fingers and position a small pair of scissors just above them, so the blades of the scissors are around the metal insert of the coloured rod. Roll the straw between your fingers while applying successive pressure with the scissors until the straw is fully cut. Remove the cut part of the straw. Grab the coloured rod to remove the rod and capillary. A wire stripper can also be used to open the straw. Positioned around the coloured rod and its metal insert, the stripper allows in one single move to section the straw around its full periphery and remove the cut part. The coloured rod is thereafter accessible and the rod and capillary can be removed gently from the straw by pulling vertically. Retrieve the biological material by immediately putting the extremity of the gutter in contact with the thawing solution.